but the variance between genes are different. It's not fair for the genes to be identified, right? Therefore, it's easy for us to identify some genes, but not for other genes, depending on their variance, right? Therefore, to make it, let's say, fair for genes, so we need to perform the VST, the variance stabilizing transformation. Okay, but this process usually is just a function f. Okay, function f for all the data we observe. For example, the counts from the sequencing. For example, the FPKM from the sequencing. So we want to have a function f to transform all our data into y. And x might have very variable variance, but y will have relatively stable variance. Okay, to make sure the data is easy to handle later on. Okay, it's also kind of pre-processing step. Okay, to make sure our data is friendly to most statistical tools. Okay, so one of the typical VST method is using a very simple approach, very simple function f is the square root, right? Square root. So let's say if your data x is a positive number, for example, the counting number counts, right? The read counts. Then simply you just take a square root. You get a y. Y equals square root of x. Then there is actually theory to tell you that so the variation of the y will be much more stabilized compared to the x itself. Why is that? Because people actually have a lot of assumptions on the read counting data. One of the most simple and also widely used assumption on the counting data is we assume that the reads counting follows a Poisson distribution. Okay, Poisson distribution. Not sure how many of you know Poisson. Anyone do not know Poisson distribution? Okay, that's very simple. That's a very simple example for Poisson. The most famous example say, let's say in one afternoon, for example today afternoon, say you just go to our supermarket, okay, supermarket. And you stay there, you watch how many people enter the supermarket, okay? So in a given period of time, for example, you watch for 1 hour, okay? And you count how many people arrive at the supermarket. Okay? You count how many people arrived at the supermarket. The number of people arrived in that period of time follows a Poisson distribution. Okay? You can think about that. So the number actually, if you put the number on the x-axis, okay, number could be 0, right? You can be there for 1 hour, no people come, right? It could be 1, could be 2, could be 3, so on, right? So then you do this experiment for many times. Then I'm asking, so how many times you see 3 people coming in that period of time? Okay, if you see, you watch 10 times, out of 10, 5 times, there are 3 people there. You put a dot here, right? And similarly, you can give some dot. And this distribution actually is Poisson distribution. Okay, this is counting the number of people enter the supermarket, right? Similarly, for the read counting data, so we are counting the reads. It could be 0, right? Could be 1, could be 3, could be 10,000 reads in that position, right? That number, the counting of that number follows a Poisson distribution. Okay, follows Poisson distribution. And also, if you check a mathematical book, you really find out that there is a property for Poisson is the mean equals variance. The mean equals variance for Poisson distribution. Okay, for Poisson distribution. So this way, the mu equals sigma. Okay, so this is why for the gene expression data, so if we assume this Poisson distribution, then we can easily to understand why the mean value and the variance is kind of a linear correlation. Okay, so if you draw a fitting line here, you will say basically roughly the mean equals the variance, right? And this is why people think the sequencing data, when we count the number of reads in that position, we can assume that distribution is Poisson. Okay? And this is also why we want to use the VST to stabilize the variance, because variance equals mean means higher mean, higher variance, right? We also mean that our data is not regular, at least, okay? Not regular. So square root is one way to do that. After the square root, you can find out that x-axis now is the mean value. So y-axis is the standard deviation of square root of x. Okay, then you will say it's more stabilized compared to a curve like this, right? It's more stabilized, but still there is a area here, so it's quite small in terms of the variance, right? Therefore, this is not the best method. Okay, not the best method. People, especially mathematicians, help us to solve this problem by further using I'm not going to talk about details. So they make it more general. Okay, make it more general. So if you assume there is a function between the mean and variance, if the variance equals h mu is the mean, variance is the variance. Okay, h is a function between mean and variance. Okay, for Poisson distribution, h is like a mirror, right? So x equals y, right? The mu equals variance for Poisson, right? But for other distribution, this function might be more complicated, right? The mathematician told us that no matter how complicated is your h, you can find a very good stabilization option for x to y using this formula. Okay, using this formula. Okay, then if you further assume that the h mu equals I square mu square, then you can now find out that y should equals log x. Y should equals log x. Okay? And this actually, after the mathematical proof, people believe that for any kind of counting data, for a kind of like a Poisson-like data or next binomial data set, you can use the log transformation to stabilize the variance from your data set. And this is actually why Ran introduced. So for any type of data, before any processing, you need to do the log 2 or log 10 transformation first. Okay? Because of the mathematical proof, I don't have time to go through the details. Again, if you have the interest, so you could check AI. I think AI could tell you the answer. So for how to do the deduction, how to do the calculation to make sure this is more stabilized in terms of variation. But for our data set actually, so similar with Ran's code, I also run the code on my data set. Okay, my data set before the log 2 transformation, the raw data. So you can see the distribution of the data size is kind of thick. Okay, this is a thick distribution. Weird. We don't even know. So, like where is the data details, right? So we see almost all the data close to 0, right? But there are several data points. It's almost 2,500, okay, close to 2,500, but with very small number of samples or very small number of genes have this kind of high values, okay, high values. But if you do the log2 transformation of the whole data set, then your distribution will become like this. So it's more closer to a normal distribution, or it's more closer to combination of multiple normal distribution. Okay? So at least from here, you can see this is the shape. It's kind of bio-like shape, right? So it's more like a normal-like distribution. All right. So here, actually, also one thing we have to pay very special attention is when you are doing the log2 transformation, there is a big issue. It's 0, right? So how can we do the log2 0? It's impossible, right? Therefore, we need to usually introduce a pseudo count. This pseudo count is quite tricky. Okay, remember, so when you are doing the data processing, the pseudo count might change your data shape. Overall shape could be changed by pseudo count. So why we should add this pseudo count, and the principle can help us to understand what's the meaning of this pseudo count. Any idea? Anyone? I gave you a hint here already. Who is this? Who is this? This guy gave a theory to guarantee this pseudo count is okay. Who can have a guess? You want to have a guess? Who is this? No, not him. Yeah, he's famous, but not him. You think it's Boston? No. Any other? Yamo, you should know, right? Who is it? Huh? No idea. Okay. Yes. Bayesian. Yes. This is Bayesian. Because this is actually the prior. The one is the prior. Basically, based on your data, you observe 0. 0 means you didn't observe anything there, right? But Bayesian told us, based on our knowledge, it's not possible you didn't observe anything, right? Therefore, we have to use our prior of knowledge to add something there. Okay, to add things there. This is something missing in the data points. It's our experience or our knowledge. Okay, basically, this one means you have the expectation that even you didn't observe anything, in reality there will be something there. This one is that something. Okay, if you are talking about the read counts, the number of reads, then this one will be at least one read should be observed there. Okay, and if you think your data has been anomalized and your data has some other range, you could put 0.1 here, or 0.01, or some other numbers you feel proper. Okay, there's no like a guideline which number you should put here, but there is a tradition or experience. People think, okay, 1 may be good, then you put 1 here. But it's tricky to really put 1 here, okay? Because you don't have the like I would say the real knowledge, okay, to say, oh, it should be 1, okay? Basically, it depends on your prior, okay? It depends on your prior. Basically depends on your understanding of the data set. Therefore, I would suggest when you are doing this kind of processing, try multiple options. try different different parameters, different pseudo counts to make sure you really understand the structure of the data set. Okay. Right. Let's see how it looks. So after the log transformation. So remember this plot, right? So this is our mean value, and here is our variance. So the mean and variance is kind of positively correlation and almost like a linear correlation between each other, right? So after the log transformation, so we do this again to find out that. So this is x-axis and this is y-axis, right? Y-axis is the variance of the log 2 of our initial data set. So you can see the correlation now becomes no correlation, right? No correlation. If you do the fitting of a line, it's a flat, right? So basically, we remove the correlation between the mean and the variance. This is kind of variation stabilization transformation. Okay. All right, so now we understand why we should do like a VST, and now we also know why log2 transformation works, and also we should pay special attention for the pseudo count. Sometimes could be 1, sometimes could be other non-zero, like factors there, positive number there, so indicating our understanding of data set or our bias on the data set, so according to the Bayesian theory. Okay. All right, so now I'm asking another question. So say, like, if we focus on one gene, so we check what is the distribution, what should be the distribution of one gene across all samples. Let's say Ran gave you, I think, how many samples? 200, right? 175 samples, like for prostate cancer, right? For prostate cancer database. So then the question is, so if you have one gene, like expressed in all samples, okay, in all samples, so what should be the distribution? What should be the distribution? What is this distribution? Normal, right? Normal distribution. Why normal distribution? Why should be normal distribution? Because the central limit zero, right? Because there's a big number theory. Because we have a lot of cells in each sample, okay? In each sample. Let's say that cell number big, right? For example, for a given sample, I have 1 million cells. I do the sequencing. I check the expression level of the gene across all the cells, right? So the observation is actually a mean value, right? For each sample, it's actually a mean value of many cells, right? Therefore, that mean value should follow a normal distribution, okay? Follows normal distribution. This is why if we checking the expression of a so-called housekeeping gene, what's housekeeping gene? Means that gene, suppose they express in all cells, and high expression in all cells, right? Then we should observe like a normal distribution. However, for some scenarios, we do observe like let me give you one example of the non-normal distribution. This is another gene, okay? This is another gene. So don't remember the gene name, but for this gene, if you check the expression, like a distribution, you find there is a huge peak here. There is another peak over here, right? There's 2 peaks for this gene. Clearly, it's not a normal distribution, right? This is not a normal distribution. So why? The question is why? You think about that? Why this gene is a normal distribution, but this gene clearly is not a normal distribution, right? Why is that? No, I told you like a number of samples is actually a number of cells in that sample. For each frequency, the number of cells, there are millions of cells, right? So many cells there. The number is big enough, right? But it's not a normal distribution, right? Okay. It's close, but not accurate, okay? Actually, I told you the answer already. IID, okay. Who knows what's the meaning of IID? Wonder, what's the IID? Okay. Independent is correct. Identical, right? Independent, identical distributions. So this is the very important assumption for the central limit theorem. Okay, if only your distribution is identical and also your samples are independent from each other, then you can observe a normal distribution. Okay, for our scenario, all the cells is kind of independent. We can assume they are independent. Okay, they kind of independent, but for those cells, they do not have the identical. They do not have the identical like a distribution, right? Therefore, we only have the ID. I means first I mean independent, right? Independent distribution, but not independent identical distribution. If you have the normal cell and tumor cell, I think yeah, you are right. So you have normal and tumor cell, they have different distribution. You take the average, you do the mean. It doesn't follow normal distribution, right? Because it's two different distribution or multiple distribution, right? So the K is IID, okay? IID is a big assumption for normal distribution, right? Yeah, this is actually impression for your data and why sometimes you could ask, so oh, this gene follows the normal distribution, but the other genes do not. So why? Okay, this is the key reason. Because of independent, identical distribution. Then you have a big number, right? Over 30 or something, right? Then you can have the normal distribution. Okay. The next question will be, so we have the impression that, so this is a normal distribution, but can we quantify? Can we really see it's a normal or not normal, right? Can we quantify it or not? The answer is yes. There is a mathematical tool can help you to really quantify the distribution, not limited to normal distribution. Actually, it can do for all distribution. You can also share the code for this like a analysis tool as well, named the QQ plot. QQ plot, okay? Quantile plot. The quantile plot can help you to recognize the distribution not by your intuition, but by mathematics, okay? By mathematics. So here, we're going to roughly go through the QQ plot with you. So basically, for any data set, for example, the one gene expression level, that's one gene set, right? You want to know what kind of distribution are you dealing with, okay? If you want to know the answer, you need to firstly have a hypothesis. You need to have the non-hypothesis. Your hypothesis basically is it could be a normal distribution or it could be a binomial, Poisson, or any distribution you want to evaluate. Okay. For here, we can directly evaluate the normal distribution. Basically, on the x-axis, is our x-axis is our theoretical quantile. theoretical quantile. What's the meaning of theoretical quantile? Let me tell you. For example, for normal distribution, what is the 50 quantile for normal distribution? What is the 50 quantile? Or what is the median value of a normal distribution? Should be the mu, right? Should be mean mu of the normal distribution. Or what's the 75 percentile? Okay, so you can define the quantile based on this kind of counting the rank, right? For example, in our box plot, we have the 50 quantile in the middle. We have the 75 quantile, 25 quantile, 90 quantile, right? For each line. Basically, we have the quantile for any given distribution. We have the we can define 1,000 or 100 different quantiles, right? And then, for example, the mean is median is a quantile. It's 50 quantile. It's median value, right? The maximum value could be 2 quantile or 100 quantile. Okay. After we define the quantile for any distribution, then we can get a number for the normal data size. For example, our data size shown here. Going back here. So what is the 50 quantile for this data set? 50% this quantile should be in the middle, right? 3.5, around 3.5, right? Then we can make a point on that QQ plot. X-axis is our 3.5. Y-axis should be 0 if we are talking about the a normal distribution or the mu value of the normal distribution. Okay, then we got 1 point on the QQ plot. Okay, then we do this for if we are talking about 100 quantile, then we do 100 points on that plot. Okay, if we talk about 1,000 quantile, we are going to have 1,000 points on this plot. Okay, so for each dot, now you can understand. So this dot here is from the theoretical value on the x-axis. The y-axis actually is from the real value. Okay. Then, if this is a straight line, then we categorically think our hypothesis is correct. So this data set follows this distribution. Okay. Otherwise, do you see outliers like this, indicating that so the data set we are evaluating is not following exactly the distribution so we are evaluating. Okay. So this is a QQ plot. So to better understand it, I would suggest you play with the code provided by Liu Ran to check for any data set. You can collect any data set. For example, you can really collect in the supermarket data set, and then to ask whether this one follow the a Poisson distribution, right? And then on the x-axis will be the real Poisson distribution, and the y-axis will be your data set. Then you can generate the QQ plot to visualize whether so your data set really following a Poisson or follow the normal or follow a binomial, right? Okay. Right. So for our data set here, we were hypothesizing that this might follow a normal distribution. Therefore, we do the QQ plot for this particular data set, and we have this kind of figure. Okay, on the x-axis, you can see this is a standardized normal distribution with mean 0, variance 1. So this is a 50% quantile. So and this value is 3.5 for this one, right? Then they can have 100 different quantiles of both distribution. Eventually, they find out this is nearly a straight line, right? Therefore, this plot can prove that. So this distribution is nearly a normal distribution. Okay, nearly a normal distribution. All right. Any questions? Let me give you some negative control. The negative control is here. So for this gene, actually, it's not a housekeeping gene. Means that gene expressed only in partial of the cells. Also indicate that these genes do not follow the IID assumption, right? Therefore, this gene has this weird distribution. Okay, we suspect this gene might not follow normal distribution. Therefore, we perform this QQ plot and find out that this is not a straight line, right? This is a weird shape, right? Therefore, so using the QQ plot, we can show that this gene do not follow the normal distribution. Okay, this is the next. So any questions so far? Right. Very good. So now we also know that why it's not normal distribution, because it's not IID, right? Next, so we go further step. So the quantile normalization. Quantile normalization. Why we need quantile normalization? So here I give you again a very simple example. So to show why the quantile normalization is important. So in a simple data set here, I have 4 samples A, B, C, D. Okay. 4 genes, A, B, C, D. I have also 3 samples. I have sample 1, sample 2, sample 3. And then I have their counting, how many reads we observe in different genes, in different sample, right? 5, 4, 3, 2, 1, 4, 3, 4, 6, 2, 4, 2, 8. Those are numbers, the counting reads in a sample for that gene, right? This is the counting. Then ask a very simple question. Can we compare the expression level of gene C in sample 3 versus like a gene C in sample 2? Okay, this is a very fair comparison, right? So basically, I'm caring about gene C. So we observe that in sample 2, gene C have 4 reads. In sample 3, gene C have 6 reads, right? We do a comparison. We can easily see that. So sample 3 have high expression of gene C compared to sample 2. This could be our conclusion based on the number comparison, right? But if I tell you this is wrong, why? Because although the number is bigger here, but in sample 3, if you checking the ranking, okay, ranking for all genes, for example, for genes sample 3, number 1 gene should be gene D, right? And the gene C rank number 2 in sample 3. But in sample 2, gene C rank number 1. It's like you are comparing the student's score, okay? Say you are comparing math versus biology, for example, okay? Math versus biology. So maybe a person got 90, okay, in math, but got 100 in biology, okay? Can you say biology is better than math? You cannot, right? It depends on many factors. Firstly, how many total marks do you have in each subject, right? Secondly, how many students and what's the ranking of that students in the class, right? For example, right? In the class. Therefore, we need to think carefully before the comparison, cross samples, okay? We cannot easily use the number from sample 2 compared to sample 3. The reason is because they have different distribution. Sample 1 has a minimal 2 to 5. Sample 3 has a 3 to 8. Sample 4 have a 1 to 4, right? They have different range, different distribution. it cannot be easily compared, right? Cross samples. However, this actually happens very often in biology studies. We always want to compare the genes cross samples, right? We want to compare cancer versus normal, right? We want to compare DNA, like in maybe diabetes versus like a healthy individuals, right? We want to compare those cross samples. So before doing that, we need to do the quantile normalization. Actually, quantile normalization is one technology to allow different samples follow a same distribution. Okay, if you have two samples, they have different distribution, then before comparison, you need to think about like quantile normalization. Okay, the following will be the like a pipeline or the like a method how we do the quantile normalization. So if you want to follow it on computer, you can already provided the code, right? So you can do the quantile normalization. So you can understand the details here. Let me go through the quantile normalization with you together. So basically in quantile normalization, we need to firstly calculate the rank of each gene in each sample. So here we have sample 1. So B rank number 1, right? Therefore we have 1, 2, 3, 4, right? And this is 1, 2, 3, 4. This is ranking of sample 1. Okay. For sample 2, we have the 1, 2, 4, 4. Basically, we have the ranking 1, 2, 3, 3, right? They're the tie here, okay? They rank the same. So they're both rank number 3. For sample 3, we have the ranking 1, 2, 3, 4, right? We have this ranking of each sample, okay? After the ranking, we want to say, okay, so after ranking, can we like use the rank to directly represent the number? The answer is no, because the rank is integer, right? But this number is usually not integer. Okay, and also the distribution of the ranking is also strange. Okay, it's a category value. It's changing too much, right? Therefore, we want to go back to the values. Then you might need to came out with an idea how to really like came back to the real value from the rank, right? So basically, you want to define what's the value for rank number 1. What's the value for rank number 1? So the method, so here they are using is the following. Basically, after ranking, you can see for each sample, we have the 2, 3, 4, 5, 1, 2, 4, 4, 3, 4, 6, 8, right? Basically, we have the rank number 1, the first ranking for all samples. For sample 1, the first ranking is 2, the last one is 5, right? For sample 2, it's 1, 4. For sample 3, it's 3, 8, right? We have the rank for each sample. Then we ask, what's the meaning of rank number 1? The meaning could be either 2 or 1 or 3, okay? Then the simple way is just take an average. If we take an average of rank number 1, then we got a value 2, okay? Then we take an average of rank number 2, we got another value 3. We take an average of rank number 3, we got a 4.767, okay? And the same thing for number 4. Therefore, we have 4 numbers to represent 4 different ranks, okay? 1, 2, 3, 4, represented by 2, 3, 4.67, 5.67. These are the real numbers now, right? So based on these real numbers, we can go back to replace the rank numbers. For example, from this rank number 4, we got the 5.67. Rank number 1, we got 2, right? Rank number 1, got 2. After this replacement, so the data still keep the range of the original data. And also, this data of each sample will follow roughly the same distribution, okay? This is a interesting like interesting method. There's one thing we have to pay special attention is remember we have one tie here. Okay, for sample 2, we have 1, 2, 3, 3, right? If we replace this 3 with the same number, 4.67, then overall for sample 2 it's not fair, right? For sample 2, it's kind of underestimated, right? Because there actually you can say it's 1, 2, 3, 3, or you can also say it's 1, 2, 4, 4, right? Because they are tied, they are both number 3 and number 4, right? Therefore, for this particular scenario, we will take an average. Okay, we can take an average between number 4 and number 5. This average value will be 5.17. We put it here. And this will be the final quantile normalized values in our database. Okay? It's clear? Good. So after quantile normalization, if you check the properties of the database, you will find out that the mean value and the max value of all the samples, they are almost the same, okay? And also the median, the mean, and the different quantiles also the same. Basically, all the samples now, they align together. They become the same distribution, okay? They become the same distribution. And based on this normalization, if we ask the question again, if we compare like a sample 2 versus sample 3 in terms of gene C, you find out that C now becomes 5.17, okay? And in sample 3, it's 4.67, okay? It's the opposite, right? Remember, at the very beginning, it's 6, 4, but now it becomes 5, 4, right? So therefore, based on this quantum normalization, C3 should be less than C2, okay? The conclusion changed based on our quantum normalization, okay? Therefore, the quantum normalization is actually very useful and important for us to compare cross samples, okay? Cross samples. All right. So furthermore, I think another normalization is also important for us to compare between genes. So remember here, for this quantum normalization, it enables us to compare sample 2 versus sample 3, right? You might have another idea or question. Can we compare gene A versus gene B in one sample, right? Can we compare 4 versus 1 in one sample? The answer is similar. If you direct compare the value, this will be sometimes misleading because different genes might have different scale. I think the example is showed here earlier. Example is actually this one. For this gene, if you compare gene A versus gene B, you will always see gene A is smaller than gene B, right? Because gene A have much small scale, small mean compared to gene B in general, right? Therefore, sometimes we don't want to see the absolute value. We want to see the relative value. How can we do that? The method is named Z-score, or scaling process. Basically, you want to scale your data into a standardized normal distribution by this formula. It's a very simple formula. X is actually your value. For any value, you minus your mean value divided by your standard deviation. Okay? Then you got a Z-score. this Z-score is not for the statistical testing. This one is for the scaling of your data set. Scaling of your data set. So if we go back, to the this here. Let's go back to the data here. The scaling process for gene A will be for all the values here, you minus the mean value 4. 4 is a mean, right? For example, 2 minus 4 minus 2, 0, 2, 0, right? So then this gene A will be coming to the mean of 0, okay? Similarly, for gene B, you use the value here, minus 400, okay? Minus 400, this 0, and this becomes 20, 10, minus 20, okay? This is a scaling process. Then after scaling, then you compare the gene expression. Then the gene and gene will be comparable because they are relatively the same scale, okay? Relatively same scale. And scaling process also being provided by Liu Ran, so in his code, so you can do easily scaling. Let me show you what the performance after scaling. So basically for our samples, remember our 20 genes, so we have very different expression level across different samples. After all those pre-processing, okay, we can make sure that all the genes follow the kind of a normal distribution and also have very similar mean value and also have similar standard deviation. Standard deviation is actually represented by the width of the box, right? So basically, they look similar distribution. Then you can compare cross samples and cross genes, okay, after this pre-processing. All right, this is Z-score normalization. So we already introduced many different steps and seems very comprehensive and give you a very rough summary is the following. Basically, for any kind of RNA sequencing data, you firstly will get the FASTQ file, right? So after mapping, you got the BAM file. Then you can counting the reads for different genes. You got the read counts, right? So based on read counts, you can do some normalization to generate the FPTM. So after you got the FPTM, so you need to stabilize the variance by either square root or log transformation. So log transformation is more recommended because it's theoretically proved that. So for many distributions, log2 transformation can give you a very stabilized variance. So VFT, after VFT, you want to do the quantile normalization to make sure between samples is comparable. Then you want to use the Z-score transformation scaling to make sure that between genes they are comparable. Okay, so this is basically the rough steps of gene expression analysis. But here I have to emphasize that so those steps they are not fixed. Okay, it's not fixed. So whether or not you want to perform quantile normalization, scaling, or VFT is heavily depends on the downstream task. Heavily depends on your downstream task. Okay, different tasks might have different choice of the steps. Okay, sometimes you don't need to do the FPM. If you use the software to generate the volcano plot. So I think Liu Ran, what software you are using? The volcano. Which software you are using? Or t-test. You're using t-test or t-test, right? Right. For different testing, you need to check details. What's a null hypothesis? Okay. Based on the null hypothesis, you can determine which step you need to choose. Okay. All right. So there are something else. So I didn't cover the details. So in the processing of RNA sequencing data, is removing of so-called batch effect. Batch effect. So you might hear this word often if you go deeper into this field. So for many type of analysis, so batch effect exists, especially for the sequencing data, RNA sequencing data. So here I give you very rough history of the batch effect correction. So from the very old data set, for example, McCree, so people start to develop method to overcome the batch effect. So using a linear mixed model or some other models to remove the confounding factor. So in your database, for example, sometimes so your data might be like a misleading because of the data generated from different labs. Okay, for example, the RNA sequencing data generated from my lab might be significantly different from the RNA sequencing data generated from another lab. Okay, so the reason is not because of the sample is not good. The reason is because in the pre-processing steps, so there might be slightly different. That slight difference will eventually dramatically change the value you are observing. Okay, and that kind of difference is kind of systematic. So it's not easy to be handled by scaling or quantile normalization. You need to perform such kind of more comprehensive model, for example, the linear mixed model, to learn the bias from different labs or different, even different students. Different students from the same lab sometimes have different significant batch effect. Okay. Also, sometimes depends on the temperature, depends on the antibody, depends on the timing, protocol. So many potential factors could be involved. Okay. So in 2007, actually a very famous method has been proposed named CANBAT. Okay, so it's still widely used. So if you have the issue of batch effect, you can try this method, CANBAT. So using the experimental Bayesian approach to overcome the batch effect. In 2012, another software package being introduced. 2018, so another method named the mutual nearest neighbors have been introduced for overcome the batch effect in single cell studies, because this phenomena of the batch effect is more dramatic in the single cell studies. Okay. Also later on, some other methods, so using dynamic programming or some others have been introduced. So if you have the data from multiple batches or multiple labs, so I highly recommend you to use some batch effect correction approach to overcome the issue from the batch effect. Okay. So lastly, that should be one example I want to share with you. So I think Ran already shared with you a more comprehensive database. So therefore, I'm not going to go through it here. So, but if you want to test by yourself, so I will highly recommend you to follow the steps. So in the following examples. So let me briefly go through with you. Then later on, you can test by yourself. Okay. So basically, for any type of gene expression analysis, usually we firstly want to define condition. So here I show you a heat map, and the color represents the expression level of genes. Okay. The rows represent the gene names. The columns indicating samples. Okay. Oh, so sorry, the other way around. The columns are genes. The rows are samples. Okay. Columns are samples. Sorry. This is the column. Okay. These are gene names. The probe names. Column. And here are one sample, second sample. Okay. Each is ID is a sample ID. Okay. Okay. So in any type of study, you could have either 2 groups or 3 groups or multiple groups. In this example, I have 2 groups. One group is samples treated by a drug treated samples. Okay. Another group is not treated by a drug. Okay. I want to understand what is the impact of the drug on different genes. Any genes could respond to the treatment. This is my scientific question, right? To answer this question, I could perform this kind of RNA sequencing analysis. In this scenario, I have 2 types of samples, 2 categories. I want to compare for any gene, for example, gene number 1, whether the gene are high or low or no difference between treatment, non-treatment, right? From the image, from this figure, you might already tell that, so this is more green in the treated samples, right? In the treated sample. Therefore, the green means low expression. Low expression. I don't have the legend here, but I tell you, green means low expression, red means high expression, right? Then you can tell from this figure immediately, so the drug might inhibit the expression level of the gene 1452475. Okay, that's the inhibition of the gene by the drug. Okay, based on the data. So basically, I want to perform the differential expression analysis for each single gene. Okay, for each single gene. For each gene, I can do a testing, right? What test you can choose? T-test, right? For example, or Z-test, right? This can use to compare two fruitful samples whether the mean is significantly different or not, right? So I think Ran also have some code for t-test, right? I think t-test. So you can do the t-test to see whether it's significant or not, give a p-value, right? So I give you some code in another alternative way in MATLAB, so you could go through this step. You log into the VDI UST8K on your computer, this is actually work computer on campus. So you can then load the database, and then you try to calculate the p-values. So using this t-test, okay, so I give you a code here already, t-test to see the treated and untreated data set. Okay, then you can got the p-value for each gene in this setting. Okay, this is the for t-test. Another alternative method, so it's named as the rank sum test. Rank sum test. So remember we introduced the Pearson correlation and the Spearman correlation, right? The difference between Spearman and the Pearson is Spearman correlation is based on the ranking of values, right? But the Pearson is based on the real value, right? So here, actually the rank sum test, also named as the U-test or very consistent rank sum test. So many different names. But anyway, this test actually is based on similar idea. It's not like evaluating the real value. Instead, this testing is evaluating the ranking. Okay, therefore, so people believe this type of testing is more stable. Okay, you can also use this one to generate p-value for your samples. So basically here, instead of a t-test, you can change it to rank sum. Typing rank sum will be a rank sum test. So right, so I will suggest you guys, when you go back home, you can do the following testing. So run this code, okay, run this code to test. So basically, if you use the real data, okay, versus random data to generate the p-value, and then check how many genes are significantly different between the two conditions, okay? Tell me the answer, okay, next week. So I will randomly ask some of you, tell me the answer. So how many genes will be significant? Compare real data versus random data, okay? Versus random data. I have all the code here. Maybe you need to do a little bit of debugging, okay? Or you can use the Ran's code, it's also okay, right? Basically, you need to generate a random distributed database and then test how many genes are significant, okay, between the two settings, okay, and tell me how many, okay. Then I will further introduce the idea behind it, so next week, okay. So lastly, so the take-home message for this week, so especially for this lecture, so basically, so we learned about the expression analysis, basic idea. So why we need to do the stabilization of the variance, right, for different variables. So basically, the higher variable generally means that more samples and greater effort are required to obtain reliable estimates or reliable results. Okay, so therefore, different variants in different genes might cause problems. Okay, you need to stabilize the variant. Okay, variant stabilization is important for most of our downstream analysis. So I suggest do the log2 transformation. But when you are choosing the pseudo count, so be careful. So because this is actually your prior or your understanding of your own database. Okay. So although we introduced according to the central limit theorem, so all the sequencing data, especially for the box sequencing data, they should follow a normal distribution. But we do see many genes don't follow normal distribution. The reason is because they are not IID, right? They are not like an independent identical distribution, right? So the gene expression analysis have many steps. So we don't need to follow exactly the same pipeline, but we do need to understand before performing any type of scientific analysis or generating any type of conclusion, we need to see which steps are required. We need to make a judgment, make a decision. So how we should do the pre-processing of database. Okay. So lastly, the high throughput data analysis. So like the differential expression analysis across all the genes. If you have 20,000 genes, you can do this testing for 20,000 times. Okay. So the reason I'm asking you to do the experiment by yourself for the random data set is because so you might see some surprise. Okay, you may see a lot of wrong discoveries. Basically, we name it technically as the FDR, false discovery rate. Okay, in the field. So basically, next week I'm going to share with you so what's the meaning of FDR and how to overcome the issue from FDR, false discovery rate. Okay, then later on, so I'm going to introduce like a more like a in-depth analysis for results from the gene expression. Okay, also in next week. So also, so just one note. So I think today Ran shared one message announcement, right, about our assignment 2 posted. Okay, so read carefully. If you have any questions, so come back next Tuesday. So we will have a Q&A and discussion next Tuesday to help you understand better about homework 2. And if you have any questions, issues, just raise your question during the tutorial next Tuesday. Okay. All right. So see you next week.
And the deadline of the homework is 1:30 p.m.
Next Thursday, right?
Yeah, next Thursday.
You have 1 week for finish this homework. Again, it's not comprehensive homework, so just finish it, submit it on time. Okay. All right. See you.